Hello students, we are still discussing transport in plants, but for now we are looking at factors affecting the rate of transpiration. First, let us begin with the disadvantages of transpiration. Transpiration leads to excessive loss of water if unchecked. This excessive loss of water leads to wilting and eventually death if water is not available in the soil. Thus, the plant is likely to die in case of water shortage. However, there are some beneficial effects of transpiration in that it helps in replacement of water lost during the process. So, as more water is lost, there is a force or forces pushing the water up. So, water is replaced. Mineral salts and water are transported up the plant in the process. So these mineral salts are dissolved in the water, in the soil water, and they are transported as water is being lost. Transpiration brings about a cooling effect of the plant, especially in hot weather. Transpiration is responsible for turgor in plants, especially those plants having weak stems. So they depend on water to be turgid or strong. Transpiration removes excess water, especially in aquatic plants, those plants growing in wet habitats. So we go to factors affecting transpiration, and they are grouped into two: environmental and structural. Environmental factors are those that are around the surrounding. That is, they include temperature, humidity, atmospheric pressure, among other things. First, let us look at how temperature affects transpiration. Remember, high temperatures increases the internal temperature of the leaf, which in turn increases the kinetic energy of water molecules. And if the kinetic energy of water molecules increases, that means evaporation has increased. So just to remind you, the kinetic energy is that energy in motion. So these molecules of water are moving in the process. They are moving up as they are being absorbed from the soil through the xylem up to the leaves. So this kinetic energy is energy in motion. High temperatures dries the air around the leaf surface, maintaining a high concentration gradient. And remember, a concentration gradient is difference between the region of high concentration and the region of low concentration. So if there is a high concentration gradient, that means more water vapor is lost from the leaf to the air. In low temperature, the rate of transpiration is reduced because there is low concentration gradient. The other environmental factor is humidity, and the rate of water loss is high in dry atmosphere due to high concentration of water vapor in the intercellular airspace of the leaf than in the dry atmosphere. This makes water vapor to diffuse out of the leaf into the dry atmosphere. The higher the humidity of the air around the leaf, the lower the rate of transpiration. And just remember, humidity is the amount of water vapor in the atmosphere. So the humidity difference between the inside of the leaf and the outside is called saturation deficit. In a dry atmosphere, the saturation deficit is high, and at such times, transpiration rate is high. On the other side, in high humidity, saturation deficit is low, hence lower transpiration rate. Under such conditions, plants secrete droplets of water through specialized pores called the hydrathodes. The process of water loss, this process of water loss through these specialized pores called hydrathodes in terms of droplets of water in hydrophytes is called guttation. And remember, hydrophytes are those plants growing in water or wet habitats. The other factor is wind, and wind is moving air. Wind carries away water vapor as fast as it diffuses out of the leaf. This prevents the air around the leaf from becoming saturated with water vapor. So, when we say something is saturated, it means it is full to capacity. So it has a lot of water vapor. And when we are talking about saturation deficit, that means capacity, or actually deficit is shortage and saturation is capacity. So there is shortage in capacity. So as the wind carries away the water vapor as soon as it diffuses out of the leaf, that prevents the air around the leaf from becoming saturated or having high capacity of water vapor. And on a windy day, the rate of transpiration is high because wind carries away water vapor immediately. On a less windy day, the air around the leaf becomes less humid, decreasing the saturation deficit, thus decreasing the rate of transpiration. So the rate of transpiration increases as the wind intensity increases. The other environmental factor is light intensity. And when light intensity is high, more stomata open, hence high rate of transpiration. Remember, most of the plants open stomata fully in daylight because the guard cells, which are responsible for closing and opening of the stomata, contain chloroplast, which synthesize glucose. The glucose is osmotically active. That means they will require water, or the guard cells will draw in water from the neighboring cells. And when the guard cells draw water from the neighboring cells, they bulge and open. That is in daylight. Water vapor diffuses out at a higher rate when the stomata are fully open than in dim light when the stomata are partially opened. The other one is atmospheric pressure. The lower the atmospheric pressure, the higher the kinetic energy of water molecules, hence more evaporation. And most of the plants at higher altitudes where atmospheric pressure is low have adaptations to prevent or to minimize excessive water loss. The other one is availability of water. And the more water there is in the soil, the more the water is absorbed by the plant. And hence, a lot of water is lost by transpiration. Transpiration depends on the walls of the mesophyll cells being thoroughly wet. So we want to investigate the rate of transpiration from the leaf surface. What are the requirements? A leafy shoot, anhydrous cobalt-2 chloride paper, glass slide, elastic bands, stopwatch, sellotape, and a pair of forceps. So the procedure is that a broadleaf is selected from a potted plant where 2 large papers of anhydrous cobalt-2 chloride are placed on both sides of the leaf, that is the upper and the lower. This anhydrous means it has no water. Then the cobalt-2 chloride papers are placed with or are covered with glass slides and time noted taken by both papers to turn pink. Another covered piece of anhydrous cobalt 2 chloride is exposed to air to act as a control experiment. What are the observations expected? The cobalt 2 chloride on the lower surface of the leaf, this one, turns pink within very short time, while the one on the upper surface takes a longer time to change the color to pink, to pale pink. Reason? Do you know why? The reason is there are more stomata on the lower surface of the leaf than the upper, especially in xerophyte and terrestrial plants. So the anhydrous cobalt 2 chloride is blue in color. When it comes in contact with water, it becomes hydrated and changes color to pink. So because there are more stomata on the lower surface of the leaf than the upper, more water is released on the lower side than the upper side of the leaf. So you also want to look at another experiment of investigating the rate of transpiration by a leafy shoot. So these are the requirements. The procedure is that the photometer is just set, as you can see here, where a herbaceous plant or a plant with a weak stem is placed or is kept underwater inside a basin. And then the photometer assembled, just as shown. And then this plant is just fixed and cut. We said we are cutting underwater so that to prevent blockage of the channels of water or the xylem vessels by air. And then a rubber band is fixed. So after fixing the rubber band here, then the plant is just connected and applied jelly or oil to make the system airtight. That is to prevent entry of air. Then this tap is opened to expel or to remove any air in the capillary tube. And then one air bubble is introduced. As you can see, there is one air by lifting the capillary tube from the beaker for a few seconds. And then there is a means of measurement behind the capillary tube so that it can measure the rate of movement of air bubble per unit time. So we want to look at the structural factors affecting transpiration. They include the cuticle, and plants growing in arid or semi-arid areas have leaves covered with thick waxy cuticle that is waterproof to reduce excessive transpiration. Such plants include the sisal, cactus, and aloe vera. Plants growing in wet habitats have problem of accommodating too much water in their tissues, therefore have no cuticle, and those who have very thin layer of cuticle on their leaves. That means the rate of transpiration is high so that it can get rid of excess water. The other one is stomata, and the more the stomata, the higher the rate of transpiration. So those plants growing in dry areas are called xerophytes, and they have few stomata to reduce excess water loss. These xerophytes also have sunken stomata to prevent the rate or to reduce the rate of transpiration as the water vapor accumulates in the tips, thus reducing transpiration. Others have stomata on the lower leaf surface, hence reduced water loss. But for plants in wet habitats, they have more stomata on the upper surface of the leaf so that they can get rid of excess water. So many plants close their stomata during hot, dry, sunny day to reduce the rate of transpiration, and that is what we call midday closure, which protects the plant from wilting. Some desert plants have what we call reversed stomatal rhythm, that is closing the stomata during the day and opening during the night when the atmosphere is cool. This helps them to conserve water in their tissues. We also have leaf size and shape, whereby plants in wet areas have large surface area for transpiration. Xerophytes have narrow or needle-like leaves to reduce water loss. If you have ever seen cactus, the leaves are needle-like. The photometer can be used to determine transpiration in different environments, as we had seen. Another factor is hairy leaves, and some plants have their leaves covered with hairs that trap a layer of still moist air on the surface of the leaves to minimize transpiration. The other one is glossy surfaces, that is, or shiny surfaces, to reflect light landing on the leaf surface and reduce the internal heating of the leaf and stomatal opening, hence reducing the rate of transpiration. So we have come to the end of the discussion. Finish this assignment. Goodbye.